Good morning, everybody. Good morning, and welcome to yet another supremely amazing lecture on the same chapter of class 12 that we were constantly dealing with from yesterday onwards, that is, strategies for the enhancement in food production. And today we are going to deal with a brand new topic of the chapter, that is plant breeding for disease resistance. Clear? Let's move on. So this is an amazing Telegram group where daily discussion, polls, quizzes, and OMR examinations are being conducted. So those who haven't yet not joined our small family of Telegram, join at BioPoint New. Those who haven't yet not subscribed our wonderful YouTube channel of BioPoint New, don't forget to subscribe our channel and don't forget to share it with your friends also if you like this video. And right now, hit the like button and support your family of BioPoint Neet. So let's begin with this chapter of strategies for the enhancement in the food production with the topic of plant breeding, which was our topic for this day. Yes? Good morning, Nila. Good morning. I hope I'm audible to everyone. The screen is very clear. Give me hearts on the screen then. Yes. So we are beginning with the chapter, the topic of plant breeding. Clear? Yes. Plant breeding, it is actually the manipulation. What do you mean by manipulation? The changing or alteration. Manipulation of the plant species to create a desired plant suitable for better cultivation, better yields, and disease resistant. So we are modifying a particular plant. We are modifying a particular plant in such a way that we get the desired plant with a suitable character such as better yield, better productivity, disease resistant, etc. Suppose you are combining, the plant breeding involves a crossing of the plants of a disease resistant plant. Particular plant A, this plant is named as A and this is labeled as B. So this plant A is disease resistant and the plant B is flavor better. That is high flavored plant. When you are manipulating the DNA or making out a new breed by combining the genes of these two plants, you will get a plant which is of the high quality, combined quality of the two different breeds, that is the disease resistant and the better flavor. So you will get a suitable plant or the product from this particular product, which is known as the hybrid variety, which is known as the hybrid plant or the hybrid variety. Yesterday's class I told you green revolution was concerned for the plants, especially wheat, rice, etc. The development was flourishing of the agriculture and it was dependent on the plant breeding. The classical plant breeding involves the hybridization of the pure line and the artificial selection to produce the desirable traits. And now we are using certain types of biotechnological tools, that is molecular genetics tools, for understanding further about the importance of plant breeding or to carry out the sequence of plant breeding. Next is, what are some of the desirable traits for plant breeding? What are some of the desirable traits for plant breeding? First one is increased crop yield. You can increase the yield and quality of the food that you get. Second one, you can increase the tolerance to certain environmental stresses, for example, salinity, extreme temperature, drought, all these things. You can increase their tolerance to all these different kinds of stresses. Third one, you can increase their resistance to insect, pest and pathogen, which in detail you will study in the chapter biotechnology. How you can make disease resistant crops in that chapter. Here also we'll be discussing about certain small things. Clear everyone? Desirable traits for plant breeding: you can increase the crop yield or quantity or productivity of the crop. Then you can make the plant desirable by what? Tolerance, making it tolerant to environmental stresses such as climate changes, drought, salinity, acidity, all these things, and increase the resistance to plant pathogens and insect pest. So all these are the different desirable traits which are required for a plant breeding method. There are certain main steps in the process of plant breeding. So we have to study the main 5 steps involved in the plant breeding, and this is a previous year MCQ. They will be asking you which of the following is the correct rearranged order of the main steps involved in the plant breeding method. This is a previous year Neet MCQ question, and those who are preparing for your CBSE board, 5 mark question as the 5 main steps, 5 marks point. I'll be dealing with the important points also in the upcoming slides. First one, collection of the genetic variability, evaluation and selection of parents, cross hybridization between the selected parents, selection and testing of the superior recombinants, and testing, release and commercialization. That is, you are giving, after testing and everything, you are giving out it for public use. After testing it by the authorities, you are giving it for the public use. So these are the 5 main steps which are involved in the process of plant breeding. So first you have to collect the genetic variabilities of that particular plant you want to breed it with. Then you have to evaluate and select the parents, the high yielding things you have to take, right? The parents should have a particular character, so that only they can be chosen as a parent, or need for plant breeding is there. So evaluation and selection of parents should be done, and cross hybridization, you have to cross pollinate or self pollinate between these two selected parents, and you have to select the superior recombinants or the progenies that are formed after this particular process. And the last one, after testing it by the authorities and release it to the commercial farms, you have to make it commercialized. That is, you have to give it to all the farmers of the country or the state or whatever, just making it as a commercialized product. First is the main steps, that is first one, collection of genetic variability. In wild relatives of many crops, a pre-existing genetic variability is available. So in wild varieties of many crops, there are genetic varieties are available. So collection, you can collect and preserve these wild variety species, and even you can preserve relatives of the cultivated species. The entire collection of plants or seeds having all the alleles of the gene in a common crop, you will call it as the germplasm. Previous year AIMS MCQ question. AIMS 2003 and AIMS 2004. Previous year MCQ question. CBSE board question also. What is germplasm collection? It is the collection of the entire collection of plants or seeds having all the alleles of a particular genes in a given crop. You will call it as a germplasm collection. Clear? Second step is the evaluation and selection of parents. The germplasm which is obtained is evaluated for identifying the plants with the desirable characters. The selected plants are multiplied and are used for hybridization. Pure lines are created wherever desirable and possible. So pure line means same as that of the parent or the ancestral generation are being created by the second step, that is evolution and selection of parents. Third one, you have to have a cross hybridization among the selected parents. So it is the process in which the desired characters are generally combined from two different parents to produce a hybrid plant. For example, high protein quality of one parent is combined with the disease resistance of another plant. Okay, so guys, see the cross A plant and B. The plant A, what you can say about the plant A, it is high yielding variety. High yielding variety, and plant B is highly disease resistant. Highly disease resistant. So when you are crossing between these two, you will get a particular progeny. Okay, that progeny will have both the characters, that is, it will having the high yield and it will having a high disease resistance also. So this particular progeny which is obtained between the breeding between two parents is known as the hybrid variety or the hybrid plants. Clear? There are certain limitations for the cross hybridization among the selected parents also, because it is a very time consuming and a tedious process. It's a very time consuming process and needs or consumes a large amount of our precious time. Hybrids may not combine the desirable characters also. Even though we have done the cross, when we will study about genetics, we'll clearly be able to know sometimes there are chances of getting the recessive alleles also. There are chances of recessive genes or alleles also can get expressed in the hybrid variety. So always a hybrid will not get being formed with the combining characters or the desirable character. Okay. Next is the selection and testing of the superior recombinants. It is crucial to the success of the breeding objective and requires careful scientific evaluation of the progeny. It yield plant that are superior to both the parents, that is both to the male and the female parent. Superior to both female and superior to both the male parent also. They are self-pollinated for several generations till they reach a state of uniformity, that is homozygosity, so that their character will not segregate in the progeny. So they are self-pollinated. So the plants or the progenies are self-pollinated until they disobey the law of segregation proposed by Mental. Actually, there is no connection over in this time, but when it comes to Mental, he has already proposed all those things. Okay. So here you have to study that they are self-pollinated for particular generation till they reach a state of uniformity or homozygosity, so that the character will not segregate in the progeny. The last one is the testing and release and commercialization of the new cultivators. The newly selected lines are evaluated for their yields and other agronomic agricultural economic, that is agronomic traits of quality and disease resistance. This is done by growing them in research field. After getting producing the seeds, you will grow them in the research field. Look, this is not a farmer, but this is a researcher. So the research, wherever you an agricultural research institute will definitely have a cultivation land also along with it. So that if they produce a new set of seeds, hybrid varieties or seeds, they will first grow it in their own farm. After that only they will make it commercialized. Because the farmers or the cultivators should not complain about it. Okay, so first they will cultivate it their own land plants, and they will yield, they will make out the yield, they will cultivate it, and finally they will use it, test all those items whether they are of the high yielding variety, whether they are disease resistant, whether they have the combined characters, whether should I sell it or not. That's why we told that it's a very time consuming and a tedious process. The evaluation is followed by testing the material in a farmer's field for at least 3 growing seasons at several locations in the country representing the agroclimatic zones. The material is evaluated in comparison to the best available local crop cultivator. So you will ask a local cultivator to make it grow in their farms for a few areas for in different seasons also you will make them grow. Because you have to identify which season is making them productive, at which time they are making utilizing all those climatic conditions. Next is the wheat and the rice. Wheat and the rice. That is, the agriculture accounts approximately 33% of the India's GDP and employs 62% of the population. I told you wheat and rice, you will have the green revolution for making the high yielding varieties of rice and wheat in midst the 1960s. So during 1916 to 2000, the wheat production increased by 11 million tons to 75 million tons. So you can imagine how the green revolution was made such an impact. The green revolution made such an impact on the farmers or on the authorities, and the rice production was increased by 35 million tons to 89.5 million tons. Clear, everyone? Yes, tell me on the chat box whether it is clear or not. Yes. Borlaug, Nobel what? Norman E. Borlaug who was an international center, who was a literate who was working in the International Center for Wheat and Maize Improvement at Mexico, developed a semi-dark variety of wheat. Very, very important MCQ. Norman E. Borlaug developed a semi-dwarf variety of wheat which was like Sonalika and Kalyan Sona, which was the high-yielding and disease-resistant varieties of wheat introduced in 1963. So you have to study 1963, Norman E. Borlaug, Sonalika and Kalyan Sona. Sonalika and Kalyan Sona are two semi-dark varieties of wheat varieties that were introduced from Mexico by the Norman E. Borlaug, who was working as a literate on the International Center for Wheat and Maize at Mexico. So you have to study those terms. Some of the term I'll just highlight in this for you. You have to study this term, Sonalika and Kalyan Sona. 1963, high-yielding, disease-resistant varieties of wheat introduced by Norman E. Borlaug, and it was a semi-dark wheat variety. Taken the screenshots, everyone. Screenshots, please. Yes. Next one. Again, you have to study here also the semi-dwarf varieties of wheat was introduced. So semi-dwarf varieties of rice was also introduced, derived from IR8. What do you mean by IR8? Which was developed at the Indian Rice Research Institute. Very important abbreviation, Indian Rice Research Institute in Philippines. IRRI. Previous year AIPMT question. And the Taichung native one, which was obtained from Taiwan. These all are the different names of rice. So IR8, which was developed from the Philippine Institute of IRRI, and the Taichung native, which was from Taiwan. Better yielding and semi-dark varieties such as Jaya and Retna are also developed. So what are the things that you have to study over here? IR8, Taichung native number 1, Jaya and Retna. So these four terms, which are the semi-dwarf varieties of rice, you have to study. Match the following question for your NEET examination. Again, important question, sugarcane. Sugarcane, you can study that Saccharum barbari. Saccharum barbari, which is grown in the North India. It has poor sugar content and yield, which was crossed with the Saccharum officinarum. So Saccharum barbari, which was a mode of sugarcane or the type of sugarcane which was grown in the North India, it does not have a good yield of sugar. And what did this researchers did? They just mixed it or crossed it with a particular type of sugarcane which is grown in the tropical cane forest of the South India, which is having the thicker stems and higher sugar content, that is the Saccharum officinarum. But they did not grow well in the North India. That is why they become flourished in the tropical canes of the South India. They got a hybrid sugarcane having a desirable quality like the high yield, thick stems, high sugar, and the ability to grow in North India. Because Saccharum barbari and Saccharum officinarum was crossed together. What was the only character or trait in the Saccharum barbari? It could grow in North India and adapt to its climatic condition, whereas Saccharum officinarum couldn't grow in the North India. So since they mixed or crossed with these two genes, what happened? The offspring or the progeny that we got has the high yielding sugar content which comes from Saccharum officinarum, South Indian variety. Then the high yielding sugar content again obtained from Saccharum officinarum, which is from the South Indian varieties, and they have got the ability to grow in North India, which is only trait they acquired from Saccharum barbari. Clear? Next is millets. So hybrid maize, jowar, bajra have been developed in India. It includes high yielding varieties which are resistant to water stress also. Okay. Maize example is hybrid maize, jowar and bajra. For example, Vivek hybrid 9. All these things are example of different types of cones. Breeding for disease resistance. So the plant diseases cause crop loss from 20 to 30% or even total. So these are caused mainly, they are prevented by fungicides and bacterial sites to reduce the diseases caused in the plant. So we have to make the plan. We have to find out a way, because in this chapter we have been constantly dealing with the strategies on how we can improve one and how we can improve the other. So the disease of the host plant is the genetic availability to prevent the pathogens from the diseases. Next, some of the plant pathogens that we have to give importance over here, it is a fungal. That is rest. Rest causes brown rust of wheat, red rot of sugarcane, late blight of potato. Guys, this is a homework for you in the comment section. In the comment box, you have to identify the name of the fungus that causes the brown rust of wheat, red rot of sugarcane, late blight of potato. So 3 terms, you have to number it. 1, brown rust of wheat caused by, red rot of sugarcane caused by, and number 3, late blight of potato is caused by. Bacterial disease, black rot of crucifers. Viral disease is the tobacco mosaic disease and the turnip mosaic disease. This is the brown rust of wheat. I'll zoom it and show you. Brown rust of wheat, red rot of sugarcane, late blight of potato, black rot of crucifers, and tobacco mosaic disease and turnip mosaic disease. Clear? So that marks the end of today's class. See you in the next class with supremely amazing third part of this lecture. Bye bye guys. See you in the next class.